Hey everybody, we are going to continue on with Mendelian genetics. And these are the 6 Mendelian crosses that are possible. And so these are going to be really important to know, especially moving forward with what we're going to talk about today. So when you cross homozygous dominant with homozygous recessive, all of the resulting genotypes are going to be heterozygous, which means that all resulting phenotypes will be dominant. So this table right here is going to be really important for you guys to get to know. I'm not going to walk through every single one of these, but just keep in mind that these genotype and phenotype ratios should be pretty fresh in your head with what we're going to be working on this week. So a test cross is something that you do when you want to figure out if a dominant individual is a heterozygote or if it's homozygous dominant. And so it's a cross of a suspected heterozygote with a homozygous recessive. And so the reason that you're doing that is because when you have an individual that shows the dominant trait, you can't really be sure if it's heterozygous or homozygous dominant. So if you cross the individual with a homozygous recessive individual, if you end up getting any offspring that show the recessive trait, like any offspring at all, then you know that the individual that you were testing was heterozygote. However, if you get all dominant offspring from that test cross, then you know that the individual was homozygous dominant. And so test crosses are, like I said, important for determining genotype of a dominant individual, because you can't know just by looking at the phenotype what the genotype is. So again, if all of the offspring are dominant, then you know that you're dealing with homozygous dominant. If you have a mix, then you know that you have a heterozygote. All right, so now we're going to dive into this idea of a dihybrid cross. Di meaning two, and so here we're crossing with two genetic traits. If you took freshman bio, you have done this before. However, you did this before with a Punnett square, which you're not going to be using this year. All right, so now moving forward, you guys will not be using Punnett squares when you're trying to figure out the offspring for a dihybrid cross. But with the dihybrid cross, you do need 4 letters to code for this cross. So you're looking at 2 traits. Each trait has 2 alleles. So here you've got the example capital T, capital R, little r. So each gamete gets 1 letter for each trait. So you have 2 traits, so T and R, and then you've got the 2 different alleles. So you've got to make sure that you give each gamete 1 letter for each trait. So if we look at this example with the heterozygote, you need to make sure that you've got gametes that have a complete mix. So you've got capital T, capital R in one gamete, capital T, little r in another gamete. You would have little t, big R, and then finally little t and little r in the last 2 gametes. So now one really big thing to kind of keep in mind here is making sure that you have the correct number of kinds of gametes, because making sure that you have that is really critical to calculating the results of these crosses and crosses that you'll see later on as well. And so what you want to do is look for the number of heterozygous traits. And when you use the formula 2 to the nth power, n is going to be the number of heterozygous traits. So again, for our example, we have 2 traits and they're both heterozygous. So n equals 2, so then 2 squared is going to be 4 different kinds of gametes are possible. And these are the 4 that we just talked about. So you've got capital T, capital R, little t, capital R, capital T, little r, little t, little r. So these would be the 4 possible gametes that can be created from this genotype right here. And you would have to do that then for both of the parents in the cross. So here in this example, you have 2 heterozygotes that are being crossed, and so you're going to have the same 4 gametes for each of these parents. And if you were to draw this out with the Punnett square, you would have 4 gametes on the top, 4 gametes on the side. That's 16 different possibilities that you would be using or calculating using that Punnett square. Now the results of that Punnett square would be 9 tall red flowered plants, 3 tall white flowered plants, 3 short red flowered, and then finally 1 short and white flowered plant. And so it gives you a 9 to 3 to 3 to 1 ratio, which is a very common ratio when you're talking about Mendelian genetics. So that is the expected ratio when you cross two individuals that are heterozygous for 2 traits. So real quick, I want to talk about the law of independent assortment. And this says that the inheritance of the first genetic trait is not dependent on the inheritance of the second genetic trait. So with the example that we just talked about, height is independent of flower color. So you can be tall and red, or tall and white, or short and red, or short and white. So you can have all of the different combinations possible. And when you look at this, the ratio of tall to short is 3 to 1, just like when we talked about the pea plants last time. And then the ratio of red to white is also 3 to 1 because it's also a dominant recessive relationship. And so when you do this, the cross is really a product of the ratio of each trait multiplied together. And so if you have a ratio of 3 to 1 for tall and 3 to 1 for red, you can multiply those together and you see that 9 to 3 to 3 to 1 ratio. This is just the experiment that Mendel did. Again, we're back to peas now. And you can see that you have your P generation here, you have your F1 generation here. When the F generation is crossed, 2 members of that F generation are crossed, that is when you get your 9 to 3 to 3 to 1 phenotypic ratio. Okay, and so this is from the book. I definitely encourage you guys to take a look at it, but we're going to kind of move on. So when we're doing all of this, what we're talking about is not a guarantee of the offspring that you are going to be seeing. What we're talking about is a probability of what you could see by a certain cross. So genetics is a specific application of the rules of probability. And probability is the chance that an event will occur out of the total number of possible events. Okay, so when we say it's a 9 to 3 to 3 to 1 ratio, we are not guaranteeing that if you have 16 offspring, 9 will look like this, 3 will look like this, 3 will look like this, and 1 will look like this. What we're saying is this is essentially the odds of getting a certain phenotype. So what we're looking at here is the segregation of alleles. So here, if you have heterozygotes, for the parents, you have a half chance of passing on the dominant, a half chance of passing on the recessive for both parents. And so then when you actually do this, if you were to get capital P, that would be 1/2 times 1/2, which gives you 1/4. Again, same thing, little p, you've got 1/4. I lied. That's a big P and a little p. So that's 1/4. Another one is 1/4. And so you can actually add these two together and you will get the 1/2. And then here, little p is your 1/4 again. All right. So genetic ratios are the monohybrid ratios. Are actually the probabilities of the results of random fertilization. So what that means is we've been talking about like these 3 to 1 ratios. That means you have a 75% chance of showing the dominant phenotype and a 25% chance of showing the recessive phenotype. So what we can do with this is use the product rule. And this is the rule that we use when we do these larger dihybrid or trihybrid crosses. And so this says that the probability that 2 alleles will come together at fertilization is equal to the product of their separate probabilities. Okay, so back to the tall and red example from earlier, the probability of getting a tall offspring is 3/4. The probability of getting a red offspring is 3/4. So then if you look at the probability of getting tall and red together in one individual, it's 3/4 times 3/4, which is 9 out of 16. Okay, and so you're able to do this with all these much larger crosses, and it's going to save you a lot of time instead of creating Punnett squares. All right, so the product rule is much faster as opposed to creating, you know, a 16 box or a 64 box Punnett square. All right, so to review the product rule, Punnett squares are usually only useful if you're tracking 1 or 2 traits, but after that they become incredibly overwhelming to look at, and even more overwhelming to try and actually complete and a lot of times they can become riddled with mistakes. So the product rule is much simpler. It's a much simpler way to determine the likelihood of getting a particular result. And to use the product rule, we determine the likelihood of getting each trait individually, and then we simply multiply those probabilities together. So you want to look at each trait on its own first, determine the probability of getting that trait, and then multiply the likelihood for all the probabilities that you've been looking at. So we're going to do an example with the ball python. Here we have a dihybrid cross with two heterozygotes. So A being normal, capital A being normal, little a meaning albino, capital P is pinstripe, and then lowercase p is not pinstripe. And so the two parents that we're looking for are both heterozygotes. And so if we look at these traits separately, when we cross two heterozygote A's, there is a 1/4 chance of hatching an albino from this cross. Okay. When we look at the P's, we have a 3 out of 4 chance of getting a pinstripe. And so then what is the chance of getting an albino and a pinstripe from this cross? Now, in order to determine that, we take our 1 out of 4, multiply it by our 3 out of 4, and we get our 3/16. Okay, so there's a 3 out of 16 chance that we would get an albino and a pinstripe python from that cross. We can do this with even more traits, so we could try it with 4 traits. Here we have pinstripe, we have albino, here we have piebald, which is going to be the blue, piebald is recessive, and then hypo, which is also recessive. So piebald would be little b, hypo would be little h. All right, and so now if we try to do this, if we have parents, here are your parents right here. Okay, so this is your lecture question. I want you guys to give this a try. So you have a parent that is heterozygous for A, P, B, but then homozygous recessive for H. And then you have another parent that is homozygous recessive for the A, the P, and then heterozygous for the B and the H. All right, so you need to find the individual probabilities, and then I want you guys to use the product rule and shoot me a message and tell me what is the chance of getting an animal that is albino, pinstripe, piebald, and hypo. So you may need to rewind this and go back and write down what's dominant, what's recessive. But do this problem, you've got your parents, and I want you to tell me what are the chances of getting an offspring that is albino, pinstriped, piebald, and hypo. All right, so send me a message and let me know what you got. All right, thanks guys.